Welcome. In this video, we are going to talk about transformation in bacteria, which is also a kind of genetic transfer, genes transfer in bacteria. We've already talked about conjugation in bacteria in earlier video, and now we are going to talk about transformation. So let's start. Transformation bacteria is the second way in which DNA can be transferred between bacteria through transformation. Transformation played an important role in initial identification of DNA's genetic material. Now let's see. What happens in transformation is that if it's a bacterial cell and it has its chromosomal DNA, and if there are DNA molecules present around in the media where the bacteria is growing, so there is a chance that one of these DNA can enter into the cell, and then crossing over may take place and a transformant may be formed. Now this is transformation. This is the process of transformation. Transformation requires both uptake of DNA from the surrounding medium and its incorporation into bacterial chromosome or plasmid. So what happens in transformation is that in the surrounding media, if there are DNA molecules present, it needs to enter this bacterial cell, and then crossing over is also required so that exchange of genetic material between this DNA and this chromosomal DNA can occur. Otherwise, this DNA, this foreign DNA, will be degraded. So, incorporation of the bacterial chromosome or plasmid is important. It may occur naturally when dead bacteria break up and release DNA fragments into the environment. In soil, marine environment, this may be an important route of genetic exchange of some bacteria. So these DNA molecules that are present in the media could be added for an experiment. We could add synthetically made DNA, or synthetically add DNA fragments into the media where the bacteria is growing in an experiment, or these DNA fragments may be from a dead bacteria who have released its genetic material into its surrounding, which can be taken up by another bacteria. So let's see this one. Here this is a recipient DNA of the recipient cell, and recipient DNA is here, and a double stranded fragment of DNA is present in the media. So in the first step, what happens? One strand of DNA fragment enters the cell, the other is hydrolyzed. So we see there is a double stranded DNA present here, this one, and only one strand is entering, only one strand is entering into the cell, while this other one will be hydrolyzed and degraded, right? So this one strand had entered, and now the single stranded fragment pairs with the bacterial chromosome and recombination takes place. So it will go to its homologous area of the bacterial chromosome, and then double crossover is taking place between this foreign DNA and the bacterial chromosome, recipient bacterial chromosome. And after crossing over has taken place, the remainder of the single stranded DNA fragment is degraded. Here it is degraded with the rest of it, and the rest is here incorporated into the bacterial chromosome. So this a transformant is formed. When the cell replicates and divides, one of the resulting cell is transformed and the other is not. This is because one is when this cell replicates, one is non-transformed while other is transformed. This is because only one single stranded DNA has entered and replication has taken place, recombination has taken place. So only one of the strand, when the DNA replication will occur, only one of the strand will have that foreign DNA while the other will not have it. So one non-transformed and one transformed cell will be formed. We know about the incomplete DNA replication, right? So the mechanism of transformation. So cells take up DNA through their membranes are said to be competent. So for example, this recipient DNA that is taking up a double fragmented DNA, this cell is taking up a foreign DNA. So this is competent cell, a competent cell. It is a competent cell. So some of the species of bacteria take up DNA more easily than others, of course. And this competence is influenced by growth stage, the concentration of available DNA. Of course, if the DNA molecules are very abundant in the surrounding, there are more chances for it to be taken up. Well, if there is low concentration of DNA, there is less chance. And other environmental factors, the DNA that a competent cell takes up need not to be bacterial. Virtually any type of DNA, bacterial or otherwise, can be taken up by competent cell under the appropriate condition. So what is saying is that this DNA, this bacteria is taking up DNA, but this DNA does not require to be bacterial chromosome. It can be eukaryotic or bacterial. Any type of DNA can be taken up. The next thing is that treatment with calcium chloride, heat shock or electrical field makes bacterial membranes more porous and permeable to DNA. Right, so if a cell is non-competent, that is, it is not taking up DNA from its surrounding, so what we could do is that we can treat this cell with heat shock, calcium chloride, etc., or electrical field, right? So that would make its membrane more porous, and so that DNA will be more permeable. It will be more permeable for the DNA to enter. And efficiency of transformation can also be increased by using high concentration of DNA. Like I said earlier, if the concentration of DNA fragment is high in the surrounding media, there is more chance of it to be taken up by the bacterial chromosomes. Right. Now, gene mapping through transformation. We have understood what is the process of transformation. That is, a bacterial cell takes up DNA, a single-stranded DNA, from its surrounding media, and that DNA enters into the cell, and then recombination occurs between its chromosomal DNA and this new foreign DNA, right? So transformation occurs that way. Now, how can we map genes using these transformation experiments? So transformation, like conjugation, is used to map bacterial genes, especially in those species that do not undergo conjugation or transduction. Transformation mapping requires two strains of bacteria that differ in several genetic traits. For example, the recipient strain might be A, B, C, and the donor cell auxotrophic A plus B plus C plus. So this is the example we are talking about. So the recipient cell we can see is A minus, B minus and C minus, right? So these alleles are in, not wild type, while the donor cell have the wild type A plus, B plus, C plus. That is, they can produce these things, A, B, C, whatever the proteins they code, they can produce those proteins, while these are A minus, B minus, C minus, so they could not produce the proteins of these that are required, right? So what happens, that donor DNA from donor cell is fragmented somehow. Either the donor cell dies or we kill it and we take up its DNA and then we fragment it. Either way, DNA from the donor cell is fragmented and then it is put into the media of the recipient cell, right? So what happens, transformant 1, A plus gene is taken up in the fragment. This A1, A plus, is taken up by one of the recipient cell and crossing over takes place, and this transformant becomes A plus, right? While it was earlier A minus, so it becomes A plus, but the rest B minus, C minus remains that. This cell takes up the B plus here. This one is taken up by this cell, so this becomes A minus, B plus, C minus. This one is taken up C plus, so this becomes, this transformant become A minus, B minus, C plus. But there is a fragment here, you can see B plus, A plus. Together two genes are being transferred and the cell becomes A plus, B plus. So what it means is that genes that are close to one another, the chromosome, are more likely to be present on the same DNA fragment and recombine together. So it is showing linkage. If there is linkage, that is physically two genes are very close together, A plus and B plus, if they are very close together, so when these DNA fragments are fragmented, they will still be together, fragmented together, and they will be passed to the next bacteria, to the recipient bacteria together, and a co-transformant will be occur. So by this type of co-transformant production, we can tell that which genes are lying close by, and which genes are away from each other, right? So A and C are not being co-transferred together. A plus and C plus must be far away from each other because they are not being co-transferred, or co-transformant is formed by them. So they must be far away. There is no linkage between them. While these A plus and B plus, these must lie close together because they are being transferred together. So it concludes the rate of co-transformation is inversely proportional to the distance between genes. So if the co-transformant, that is two genes are being together transformed, together it is being transformed, then it is inversely proportional to the distance between them. So if two genes are being transferred together into a bacteria, that means the high co-transformants are formed. That means there is low distance. The distance between those two genes is very low, right? If the two genes are being transferred together in high number, that means the distance between those two genes is low. That is, they are linked together. Here we can see it more clearly. A plus is being transferred, only B plus is being transferred, only C plus is being transferred, while B plus A plus is being co-transferred together. So that means that these two genes, B plus and A plus, lying close together, they are showing linkage. So they are being transferred together. So co-transformant are formed, and this is the way gene mapping is shown through transformation. Thanks for watching. Please like and subscribe.